After library construction, we use a qPCR assay to determine our library concentrations. This step is critical in helping us hit our target cluster density without performing a titration run for each library. We set up our qPCR assay on the Agilent Bravo automated liquid handling platform. We've used both 96 well and 384 well qPCR machines and found they perform equally well. We assess each library in triplicate at a 1 to 4,000 dilution. We use the Kappa Biosystems Master Mix. We used to use the Strategene Master Mix kit, but switched to Kappa for two reasons. It has a faster runtime, and we also use Kappa's pre-made standards. We used to make our own standards, which worked fine, but it was a large effort. It's quite helpful to use commercially made standards, especially in a high throughput setting, because of the amount of quality control required for maintaining a reliable set of in-house standards. At the Broad, we've made some improvements to the cluster station. One of our inventions is a lock and key fit holder for our hybridization library strip tube. This key is unidirectional, so the strip tube holder can only be placed one way. This ensures that libraries flow through the correct lanes. Another practice we developed is parsing out our lane waste lines and labeling them 1 through 8. Illumina has incorporated this change into newer cluster stations, but if your waste lines are not labeled, then your Illumina FAS or FSE can identify and label them for you. Collecting and measuring each lane's waste volume individually can help in understanding downstream phenotypes and simplifies the diagnosis of mechanical problems on the cluster station. We use several reference and tracking sheets throughout the process to help us maintain a standardized, high-quality, high-throughput system. The Flowcell Designation Worksheet is used to keep track of a flow cell as it goes through the cluster generation and sequencing process. The upper portion of the worksheet includes details like flow cell barcode, sample name, paired or single read, library loading concentration, and number of sequencing cycles. These details are generally filled in by project coordinators who base them on project goals and the library concentrations determined using qPCR. These details are all tracked in our electronic LIMS as well, but it is helpful for the cluster generation technicians to have a hard copy to keep with the flow cell for referencing and for jotting notes. The lower portion of the worksheet allows us to keep track of what step of the process the flow cell is in, as well as user comments that might help in understanding the run results downstream. Here we have our cluster generation reagent Thaw reference sheet. It includes reagents for cluster amplification, LBPA, and read-to or paired end prep. Remember, here at the Broad Institute, we perform linearization, blocking, and primer annealing as a separate LBPA module to allow us to perform a cyber QC step after cluster amplification. We'll get to that QC step later. The reagent Thaw reference sheet is color-coded according to the colors of Illumina reagent tubes as they come in the kit. For each reagent, we include the name, and in some cases the purpose, the position numbers, the starting volume for all strip tubes except the library strip tube, and the thawing condition, which kit components should be thawed on ice and which should be thawed at room temperature. Having a quick reference sheet at each station is helpful. This one shows the expected flow volumes for the various steps in the process. For example, waste produced by lane during cluster amp is about 3 to 4 milliliters. Cluster stations may vary slightly, so you should verify or adjust these numbers accordingly. If the flow volumes do not meet the specifications you've determined, you should double-check the seating of the flow cell and manifold. If these look fine, you may have some sort of line blockage or an internal mechanical failure. We use this cluster generation checklist throughout the cluster amplification process to record very detailed information each time we perform the process. A similar checklist is used during the LBPA module of the process. Once we've filled the strip tubes, we weigh them both before and after use. We visually inspect the reagent tubes before and after use, recording their lot numbers and checking for proper consumption by using the gradations on the tubes. We also check the waste produced by lane by weighing the 15 milliliter waste tubes before and after the process. The quality checklist at the bottom of the page is where we note information about the condition of the flow cell and the performance quality of the cluster station. We also check for proper priming, flow, thermal profile, and manifold functionality, and make comments about anything that might help us later. The information on this page can be extremely helpful when we need to troubleshoot downstream failures. If you notice improper, poor, uneven, or slow flow on the cluster station, it might be due to small clogs in the lines. A simple solution is to try to flush the clogs away using the bridge manifold. You can set it up to bypass the Flowcell without having to remove the Flowcell from the cluster station. After pausing the recipe, unhook the two ends of the amplification manifold, but keep the Flowcell clipped into its environment, undisturbed. Attach the wash bridge. In the cluster station software, select Reagent 13, or any reagent position that just contains water. Pick a relatively high flow rate of about 300. Don't set the rate greater than 500, because that can wear out the syringes. Next, select a relatively high volume, such as 300 microliters, and hit Enter. This high-pressure, high-volume flow should force any small clogs through the lines, going through the bridge manifold and not through the Flowcell. As we described in the classroom presentation, we perform a QC step after cluster amplification, before linearization, using cyber green intercalating dye to visually check for double-stranded clusters. This allows us to be sure that amplification was successful and that the libraries were loaded at proper concentrations. We can also identify mechanical problems with our cluster stations. Because we're such a high-throughput lab, we have a dedicated cyber cluster station in a small darkroom with a microscope. You can also use a fluorescence scanner like GE's Typhoon Imager. The point is to be able to visualize the clusters in a standardized way. After cluster amplification, we move the Flowcell to the cyber cluster station. We flow cyber through the Flowcell, followed by scan mix. We then place the Flowcell in the microscope, which is fitted with a xenon bulb and an FITC filter to allow the cyber signal through. Using a 40x objective lens gives us a clear view of the clusters. We have a camera and computer attached to the microscope. This isn't necessary for the QC, but it helps us with tracking. We scan the Flowcell barcode and photograph an image from each lane, and then save the images in a Flowcell-specific archive folder. This allows us to double-check the images later, if we need to. Again, photographing the lanes isn't critical, but it can be extremely helpful for tracking and for downstream troubleshooting purposes. Finding the edge of a lane is helpful in focusing. Cyber fades with exposure to light, so it's important to be roughly focused before you reach the area you want to visualize, especially if you are photographing it. The lanes are visualized one by one. With experience, it's possible to learn to estimate the cluster density by looking at the Flowcell in this way. The most helpful way is by saving cyber images from a number of lanes and comparing them to the cluster densities determined for those lanes during sequencing. We put together a reference chart to help in estimating cluster densities at the cyber step. The optimal cluster density depends on things like the project and analysis pipeline, but if you know your target density, you can use cyber to QC. Flow cells passing the QC are filled with storage buffer and sent to the LBPA and sequencing queue. Failed flow cells are reworked. Failures are usually due to improper loading concentrations, which are then adjusted for rework. Sometimes failures are due to mechanical problems with the cluster station. A failing Peltier block or thermal block can cause variations in the efficiency of amplification chemistry, affecting the final cluster phenotype and density.